Hello and welcome back to AP Psychology. I'm Miss Lee and we are still in the Sensation and Perception unit. That's Unit 3. This video will be Section 3, Vision. The learning targets we'll be covering today are to describe the vision process, to explain common sensory disorders. If you haven't done so already, go ahead and grab your notes and let's get started. Okay, so it's very important that we know the anatomy of the eye, the names of the important parts of the eye, so that we can better understand their functions in relation to each other. So let's go through each one. So first, as light is entering your eye, we see things because of light. First, the light goes through the cornea. This is the transparent outer covering of the eye. So we are starting at the left side. And if you kind of imagine yourself traveling through the eye with light, or as if you were light, this is the first item that you're going to kind of come into contact with. This is the transparent protective covering of the eye. It's clear. And then you have the pupil. This is the hole that lets light into the eye. It is controlled by a muscle that controls how much light enters the eye. That's called the iris. This is also the colored part of the eye. And one of the ways that I remember what the name of this part is that irises are also flowers, and flowers are colorful. So I remember that the iris is the colorful muscle that controls the pupil. It is what's going to constrict, bring in, or dilate, get larger. And that's going to control how much light enters the eye. Then you have the lens. The lens is what actually focuses the light on the retina. If you think about a camera as a metaphor for the eye, the lens is exactly that part in the camera. It's what's going to focus your picture. Well, this is what's going to focus the light on the retina. So going to the back of the eye now, you have the retina, and this is your photoreceptor area. This is what contains the visual sensory receptors, rods and cones, and we're going to get to that in just a moment. It also contains the fovea. This is the point of central focus. It contains the majority of the eye's cones, and this is where the sharpest image is going to be. Then you have the blind spot. This is where the optic nerve actually leaves the eye. Because it's technically optic nerve, there's no room for photoreceptors, so there literally is a blind spot. There's no information being processed there. It's too busy being an optic nerve. And so the optic nerve takes the message to the brain. This is the part, and you will see this kind of replicated throughout the sensory organs, but this is the part of the eye that actually sends the information to the brain for processing. Okay, so how do we actually see? We see because of wavelengths of light, and the hue, or the color, is the dimension determined by the wavelength of the light. So you've got different wavelengths, and it depends on the distance from the peak of one wave to the peak of the next wave to determine the wavelength. That's what the wavelength actually is. And if you look at the picture on your screen right now, you notice the longer waves of red are at the top, and the tighter or shorter waves of blue and violet are at the bottom. This is pretty important because this is how our cones actually determine what colors we are seeing. Different wavelengths of light result in different colors picked up by our retinas. Different cones are sensitive to different wavelengths, and so they're going to pick up different colors of light more easily than others. And the intensity or brightness is determined by the amplitude, or how actually high the wave goes. So you've got the wavelength, how long it is, and you've got the intensity, how high the amplitude of that wave is going to actually reach. That's the intensity, or the amount of energy in the wave. All right, so there are some problems in the eye. If you have misshapen eyes, it could result in different vision problems. Nearsightedness is a condition in which nearby objects are seen more clearly than distant objects because the eye is elongated. It's kind of stretched out like an oval, like more of a horizontal oval. And so the image focuses before it hits the retina. It doesn't actually land on the important part of the retina. It's going to focus in front of the retina. And this is technically called myopia, or nearsightedness. These are some examples or some simulations of what what the difference in vision actually is. The photo on the left is an example of myopia versus normal vision. Farsightedness is a condition where far objects are seen more clearly than near objects because the eye is shortened. It's squished up, and so it's more shaped like an oval vertically. The image focuses after it hits the retina, and so it's focusing behind the retina. And if you look down here at the illustration, you can see that the image is focusing behind the retina, and this is also called hyperopia. So again, you have some examples of what this looks like in real life. People who have farsightedness have a hard time focusing on items that are close to them. They sometimes will pull the item away. If they're reading a book, they'll kind of pull it away. And then that's normal vision on the right. Okay, so let's talk about the retina because this is really where the magic happens. This is the light-sensitive inner surface of the eye. It contains the receptor rods and cones plus layers of other neurons. These are neurons that assist in the process of seeing. And light will first travel to the back of the retina and then move forward. That's where it's going to hit the optic nerve that's going to send the message to the brain. So if you look down here at the illustration, the source of light is moving through the eye. It hits the back. It's going to hit the photoreceptors. In the photoreceptor, the photons of the light strikes photopigments, and those photopigments split. This causes chemical reactions that produce messages. The message is transmitted to bipolar cells. So it's going to go to a bipolar cell, and then the message is transmitted to the ganglion cell. The ganglion cell actually turns into the optic nerve, and that's where the message is sent to the brain. All right, so let's talk about the fovea. The fovea is the central point in the retina. This is where all of the cones cluster together. And because of the cones clustering in the middle, there's not very much color vision in the farthest periphery of our vision. So if you think about, like if you kind of put your hands out here, you can see them, but the colors aren't very clear, and really the details aren't very clear either. So it's because the cones clustering in the middle of your retina, so you're not able to make out the color and the detail of items in your peripheral vision versus items that are right in front of you that you're focusing on, that you're looking straight at. So let's talk about photoreceptors. You have two different kinds. You have rods and cones. Rods are sensitive to light. They're going to be able to tell you if you are in a dark room or not. Cones are sensitive to color and fine detail. So if you look at this image, notice what the next cell that the rods connect to versus the cones. What is the difference? So I want you to really pay attention right here versus right here. Can you tell the difference? So remember I told you I'd come back to bipolar and ganglion cells. Bipolar cells receive messages from photoreceptors and transmit them to ganglion cells, which then forms the optic nerve. The optic nerve is what sends the message to the brain. Cones each have their own bipolar cell. So there's the difference that I was trying to help you get to in the last slide. And so because cones have their own bipolar cell versus rods, which share one bipolar cell, the message from a rod is not sent as clearly. So we're going to do a demonstration in class to test out your blind spot. Just hang tight. Everyone has one. You just may not have ever been able to tell it. All right, let's talk about feature detectors. So this is really cool. Nerve cells in the visual cortex that respond to specific features like edges, angle, length, and movement. There are even some feature detectors that are specifically sensitive to the human face. There are parts of our brain that are designed to pick up the nuances of a human face. So let me ask you this. Would feature detectors be a sensory or a perceptual experience? What do you think? Well, because feature detectors are in the brain, and the brain is not a sensory organ, it is perceptual. Feature detectors are a perceptual experience. Visual information processing. Processing several aspects of the stimulus simultaneously is called parallel processing. So the brain divides a visual scene into subdivisions like color, depth, form, and movement. And we're going to demonstrate this by isolating your actual movement feature detectors. So we're going to watch a short video, 30 seconds, and when the video finishes, I want you to look at something that is static. Look at the back of your hand or a clear piece of paper. You're going to see something really pretty phenomenal. You ready? Just look at the middle. You should have seen something pretty fascinating. Jot down on your notes what you saw. Okay, so let's talk about theories of color vision because there are a couple of different theories about why we see and how we see different colors. The first one is the trichromatic theory or the Young-Helmholtz theory. This is based on behavioral experiments. The researchers suggested that the retina contains 3 receptors or cones sensitive to red, blue, and green colors. The kind of proof in the pudding is the color deficiencies or color blindness. This is a genetic disorder that prevents individuals from discriminating between certain colors due to a weakness in or a lack of one of the cones or more. The most common form of color blindness is difficulty distinguishing between red and green, so it's called red-green blindness. A complete color deficiency, it does exist, but it's super rare, and it would basically be like watching a black and white movie. And you see some examples of at the top, this is what an eye doctor is going to test your color blindness or your color vision by asking you what number you see here. So there should be a number emerging in a different color. If you don't have any kind of color blindness or color difficulty, you should see a number. And down at the bottom, you see kind of what it would be like if you had a red or a green color deficiency. If you had a red color deficiency, this is the apple that you would see here versus what is reality, what a normal sight person would see. And then here is what a green deficiency might look like. So this is what the apple really looks like, and this is someone who has a green deficiency would see. All right, so the other theory of color vision that you need to know is called the opponent process theory. Herring, back in the 1800s, proposed that we process 4 primary colors that are opposed in pairs of red, green, blue, yellow, and then black and white. And one of the ways that this theory is kind of proven is in after images. So basically what the idea is that if you only see one or two different colors, you're going to wear out part of the ability to see. What's going to be left is the opposite. So if you kind of look back at the colors, if you wear out the blue part of this pair, what you would see instead would be the yellow. If you wear out the part of the color green, what's going to be left is going to be red. And so what we're going to do is I want you to gaze at the middle of the flag for 30 seconds. I will time you. When it disappears, I want you to stare into the middle of the white screen and then write down what you see. You ready? Go. Okay, you should see something pretty amazing emerge. Let me know in your notes what you have seen. And that is it for vision. Our next video will cover the auditory process or hearing. I can't wait to see you then. Bye for now.